Good morning to all my dear students. In our last class, we have started a new chapter, that is biotechnology and its application. And you all have learned that how the biotechnology is providing services in the field of agriculture, medicine, food industry, power industry, etc. In our today's class, we are going to learn about the contribution or application of biotechnology in the field of agriculture. Now, when we talk about agriculture, then agriculture basically deal with increase in food production, and this food production can be increased by various methods. Now, this food production need to be increased because the growing population demands need to be fulfilled. So, there are different ways by which the yield or the food production was tried to increase. The first method is agrochemical based agriculture, the second includes organic farming or organic culture, and the third method includes genetically engineered crop based agriculture. Now, in India, the program Green Revolution was carried out in order to increase the crop production by using the improved crop varieties, better crop management practices, and by using the fertilizers and pesticides, that is agrochemicals. We became self sufficient uh, in the field of cereal crops. We started exporting them also, but these agrochemicals were very expensive and farmers were not able to afford them for a longer period. Besides that, the overuse of agrochemical, that is fertilizers and pesticides, had harmed our nature and they have adversely affected the health of the farmers also. So, the other way by which the agriculture products can be increased was introduction of genetically modified crops, and these genetically modified crops were basically cultured by understanding the genetics of the crop. The contribution that these crops have given in the field of agriculture is listed below. Now, first is the vitamin A enriched rice, which is also called golden rice. Here, the nutritional quality of the rice is increased by adding the vitamin A to it. The better nitrogen fixing crops were developed. The crops which were developed by genetic engineering, they were tolerant to abiotic stresses like cold, drought, salinity, heat. They were requiring less amount of chemical pesticides because by genetic engineering, the biotechnologists have developed the pest resistant varieties or the crops. In such crops, the post harvest losses were very less. Best example is the tomato. They have increased the efficiency of mineral usage without affecting the fertility of the soil. Many disease resistant crops were developed and micropropagation for the biomass energy production. These are all the contributions that are made by biotechnology or genetic engineering in the field of agriculture. In our today's lesson, we are going to learn about the genetically modified crops with the pest resistant varieties. Now, here is the image in front of you that is giving you the top 10 genetically modified food that we are consuming, that is rice, corn, soya, tomatoes, then papaya, dairy products, etc. So, let us start a lesson with the genetic engineering and how it has contributed in developing the pest resistant varieties or pest resistant plant. Now, pest resistant varieties, when they were developed, then it directly reduced the use of pesticides, and when the pesticide use is reduced, that means we are now not allowing a chemical to enter the food chain. One such example of biopesticide is Bt toxin. The Bt toxin is basically produced or isolated from bacteria, that is Bacillus thuringiensis, and this Bt toxin was further incorporated in various plants, and different varieties of Bt cotton, Bt rice, Bt tomato, soya bean and potato were obtained, which were having the disease resistant power towards the certain pesticides. Now this Bt toxin gene was isolated from bacteria and was expressed in plant to provide them resistance. So there was no need of insecticides for these plants. That is, the Bt was serving as a biopesticides. Now let us learn that how this Bt toxin or Bt gene is working. The Bacillus thuringiensis or Bt toxin or the protein which is isolated from this bacteria is killing the insects of particular orders like Lepidopterans, which include tobacco budworm and armyworm, Coleopterans, which include beetles, Dipterans, which includes flies and mosquitoes. This Bt toxin is basically in the form of protein crystals, and they are produced in as a protoxin or the insecticidal protein in the body of the bacteria. Now when they are in the body of the bacteria, they are inactive, so they do not cause any harm to the bacteria. But when these Bt toxins are ingested by the insects, then inside the gut of the insect is the alkaline pH, and in that alkaline pH this protoxin become activated, and as a result of this activation they start forming the swellings and perforations in the gut epithelial cell of the insect, and further the insect is dying. So that is why these proteins are isolated from the genes which are called cry genes, because they are making the insect cry by creating the perforations in their midgut. Now it is a very important question from in the CBSE board that how this Bt toxin is not affecting the bacteria and how it is creating the perforations or how it is killing the insect. So just now we discussed that since these are present in inactive form in the bacteria, that is why the Bt toxins are not harmful for the bacteria, but the alkaline pH of the gut of the insect is making them activated, and as a result it causes the swelling and perforation in the gut lining, and so the insect is basically dying. The cry genes are very specific genes and they are acting on specific pest. For example, cry 1AC and cry 2AB are affecting the cotton bollworms, and cry 1AB is affecting the corn borer. Now from the image here you can very clearly see that the Bt toxin or Bt pores are spread on the crop, that is the corn crop. When this Bt is ingested by the corn borer insect which is feeding on the corn plant, then in the gut of this insect the alkaline pH is there, and as a result of this alkaline pH this protoxin is getting activated, and it result in the swelling and perforations in the gut lining of the insect, and the further the insect is dying. So this is how the pest resistant variety is produced by using the Bt genes from the Bacillus thuringiensis. The specific genes from which the Bt is produced are known as cry genes. So this is the important contribution that is made by biotechnology by creating a new crop disease resistance or pest resistant crop. That's all for the day. Thank you.